First of all, I understand that there are people from different disciplines, so I'll tell you what I think animal breeding is all about. And we are mostly concerned with the genetic improvement of animals that serve mankind, mostly with agricultural food animals. And the question comes at what individual is best, and then we have to define a merit function. For example, in a cow, you are interested in milk production, in milk composition. You're also interested in fertility. You're also interested in health. And you're interested in lifespan. So it's a multivariate field, just by definition. And then that brings us to the next problem, is how we decide that an animal is better than another one, and this is typically formulated as a statistical problem. Then once we have genetic assessments of the merit of these cows, for example, pigs or birds or fish or whatever it is, we formulate selection programs, mating plans, inbreeding management policies, and also outbreeding. Like there are many species, like in maize, for instance, in which crossbreeding is paramount. Our data are essentially non-experimental. They used to be experimental, but it was readily realized that the animal breeding experiments needed a scale that was not manageable. So most agricultural experiment stations withdrew away from experimentation, and we have essentially relied on observational and retrospective records. These are farm records, and we have a notorious absence of randomized experiments. So in that sense, there is a lot of similarity with the sort of data that are processed in medical sciences, which are largely observational. And the inputs that we use in this statistical exercise are measurements on the animals, such as milk production, growth rates, genealogies. We normally have pedigrees, we call it, that tells you who are the parents of who. More recently, markers. Now we are in the process of obtaining large numbers of DNA sequences in animals. And there is also a beginning of usage of post-genomic data, such as gene expression and methylation. Gene expression has been used for more than a decade in the context of learning about the physiology and organization of traits, but they may play a role in prediction of complex traits. So for this reason, the animal breeding sciences are mostly agricultural giganerds. Okay? They are interested in number crunching, contrary to what you expect from the name of our field. I'm going to take the liberty of making a bit of a glossary of some terms that you may not be familiar with and may inadvertently use in the jargon. We are used to speak the same language to colleagues. So when I use the term phenotype, I refer to something that is measurable, such as the yield of a grain per hectare or the number of tumors in a patient. So for example, in a male, the prostate-specific antigen, something that you can observe. By a complex trait, we typically mean a trait in which we don't have a very good understanding of the base of inheritance, and we tend to believe that there are many hundreds, if not thousands, of genes at action, perhaps working in some concerted manner. And the trait can be measured in some continuous scale or can be meristic, for example, absence or present, or having some sort of order in a categorical scale. Pedigree, already mentioned what that is. Another word that we use commonly is additive model, in which we make an abstraction and we say the genetic value of an animal is defined by the sum of the contributions of a number of variants at a unknown number of loci. By genomic data, we most of the times refer to molecular markers or variants or sequences. Heritability is a concept that refers to the fraction of the total variability in a population that is accounted for variation in additive genetic effects. That's called heritability in a narrow sense, and that plays a role in selection programs, a predictive role. And also there is another term that I may use later on called epistasis that refers to what medical scholars call gene-by-gene interactions. But this is an oversimplification because they can be also gene by gene by gene, or gene by gene by gene by gene, or networks of interactions. Now, we talk about complex traits. Why do we think that these traits are complex? Well, from your first course in biochemistry, you know that complex processes are the result of a very large number of integrated biochemical reactions which interact with each other, and they also interact with the environment. Also, it must be true that this epistasis phenomenon, gene by gene, gene by gene by gene interaction, is pervasive. Why? Well, you just take a look to any simple biochemical cycle, such as the Krebs cycle, and you start counting enzymes, and there are many enzymes that intervene together. And if you remember, if you remember an old view of genetics, one gene, one enzyme, that involves that many genes are in action in a simple biochemical reaction such as the Krebs cycle. But obviously, if I do a knockout experiment, let's say that I do an intervention and I change the allelic variance at each of these genes, the cycle is not going to add up or subtract. It may stop, like in a knockout experiment. So interaction must be universal, and there is ample evidence from theoretical biology, regulatory biology, that it must be an essential thing in the organization of complex traits. Now, because things are very complex, we have essentially worked with abstractions. So the history that we'll review from now on is essentially a history of abstractions. So we observe Y, that's our phenotype, and the first abstraction is that we postulate there is some function of genetic and environmental functions, forces, and we do not know what this function is all about. So we have to learn that function. And until recently, most people would like to do is to propose models. And there is a zillion number of models that you can propose for any problem in question. Then it comes to the problem of model selection. You have to calibrate, assess the model, and then make stories about the output from that model. Let's start with the first abstraction relevant to quantitative genetics, which started with Galton. Galton coined the term of regression of offspring on parent. And what Galton observed from data is that when you have a group of tall parents, the children of that group tend to be on average smaller than the average of the fathers and mothers. And likewise, when you have a group of small parents, the children tend to be a bit taller. So when you observe the difference between the means of these two groups in the progeny is smaller than the difference between the groups in the parents. So there was a regression to the mean. Of course, you have movements in the opposite direction. But he coined this term, and as everybody knows, the regression is an essential concept in linear model theory, and Galton's abstraction gave an impetus for the use of linear models. And really, we have not changed very much. Animal and plant breeders, essentially and mostly, for some good reasons, used linear models. And the linear models have a good ability to describe data. Quite often, they have a good local predictive ability, but they do not provide an explanation of complexity. And you don't need to go very far. Just look at Galton's data again. This was revisited by Wachsmuth et al. In 2003 using a non-parametric method, and described that this regression of offspring parent was not linear. There was actually a bend, and the bend remains unexplained. So what is the explanation for that? Well, there must be some concealed substructure. This is the terms of the day. Everybody talks about population structure. So let's take us now to another person called Pearson, and the reason I'm doing this connection because Pearson was the first one that recognized that sometimes you observe data, but the data are heterogeneous. So he worked with crabs from the base of Naples, and this was the first time that a finite mixture model was applied to biological data, probably to data. So he postulated then there were two subspecies of crabs by looking the ratio between forehead to body length. Now, something that is largely unrecognized by my colleagues in animal plant breeding and evolution, they talk about Price's equation and the breeder's formula, is that all these equations were actually in a paper by Carl Pearson in 1903, where he said if you have a vector that has a multivariate normal distribution and you do something with a subset of that vector, then the mean and covariance matrix of the remaining distribution after you intervene we normally call that selection, changes. And that result was used by Henderson in an important paper in 1975. I will talk more about Henderson later. So Pearson had said several important things that unfortunately they remain unrecognized. Now, let's move now into animal breeding and try to say something about what we will call the founding fathers. From left to right, Sewell Wright. Sewell Wright was an evolutionary geneticist that made all his career at the University of Chicago. He retired when he was 65. He was forced to retire. So the genetics department of Wisconsin had the vision of inviting him to come and stay in Madison, and he continued working until he was 98. Actually, during his retirement, he wrote a 4-volume book. And he died because he walked in the ice and he slipped and he fractured, had a post-op. So maybe he will still be kicking around. In the middle is Ronald Fisher. He invented modern statistics. In 1922, he introduced the method of maximum likelihood. And in 1918, he introduced no less than the analysis of variance in a paper that I will make a reference to. And in the right is the most interesting of the three, Haldane. Haldane was a British geneticist, mathematician, and I will make some reference to some of his work. And there is a photo on the top, right angle on the top, where he's not lecturing genetics. He's actually haranguing the masses to organize and combat against the capitalists, because he was a Marxist. And actually, Haldane, being a British citizen, he resigned British citizenship and he took up citizenship in India. But he would spend the summers in the French Riviera. And he used to go to Hotel Negresco. So there was a professor at Iowa State University, J. Lush, that had a very distinguished career, and he put together ideas from Fisher, Wright, and Haldane, and integrated and put them in the context of food animal production, and created the foundations of the science of animal breeding. I'm told that Professor Xu here went to high school with a child or grandchild of J. Lush.
It's a small town, and there were some Lush kids at our high school, so I'm pretty sure there's a relationship.
So Iowa State University had actually the first graduate animal breeding program. More than 150 persons graduated under Lush. But you know what? He got his PhD in Wisconsin in 1922. Now, let me just highlight 3 contributions of each of these 3 giants. The one that I like the most about Haldane, because he was interested in estimating some things that we call allelic frequencies or gene frequencies, and he used a Bayesian method. So he invented something called the Haldane prior. And he argued in the following manner: if you're going to try to find mutants, let's say you go to the Amazon jungle, you organize the trip, you would not expect that there would be no mutant, or you would not expect that everybody will be a mutant. So if you go there and come up with no mutant, there must be something wrong. So he suggested that when you use the method of maximum likelihood, which you take the ratio between the number of mutants by the total number of individuals you scored, you should do something. If it is 0, it's a ridiculous estimate. If it is 1, it's also ridiculous. So shrink it or something. So he produced an unbiased estimator of allelic frequency by using a Bayesian argument. Wright introduced the concept of inbreeding and many other things. This is one of the things I'd like to highlight, which is the correlation between the additive values of uniting gametes. Then a French mathematician casted this in a probabilistic way called Gustave Mallecourt. And Fisher had 2 papers that I think are quite central in animal and plant breeding. One was his 1918 paper, between the correlation between relatives, and the second one was the fundamental theorem of natural selection. In the first paper, Fisher was 20 years old. He was an undergraduate at Cambridge. He submitted the paper, and this paper was turned down. And one of the reviewers said that it was totally useless, that he couldn't see any way in which Fisher's idea could be of use to anybody. And in that paper, he was not only setting the foundations of modern quantitative genetics, but also, as I pointed out, he introduced the analysis of variance. not a minor accomplishment for an undergraduate. In the second paper, in the second contribution in a book, actually, he suggested, there's a theorem that is a bit obscure, as most of his writings, that in the process of selection, natural selection, the rate of change of the population is proportional to the additive genetic variance at any given time. So additive variance, in some sense, is a driver of evolution in the Fisherian sense. Now, even though now we use multivariate analysis routinely, as I said, this was recognized actually quite early. And in 1936, there was an Australian plant breeder that published a paper saying, well, if you are going to select a variety of wheat, so to speak, you have to consider several things simultaneously, like yield, resistance to disease, dry matter content, etc., industrial behavior of the flower. So you have to combine all these traits in some selection index in a manner that reflects the relative economic values of the different traits, the different attributes. So he introduced this idea, and for those of you that are in animal breeding, you will recognize some of the classical formula, but I'm not commenting about those. Subsequently, and this paper has had more impact in animal breeding, although actually Smith's paper is much more elegant and much more general. Lanois Hazel, a student of Lush, introduced the idea of genetic correlation. So he postulated that when you have a couple of traits, observables, like Y1 and Y2, there may be sources that are genetic that create a correlation, and sources that are environment that create also a correlation. So we distinguish between the genetic and the environmental correlation. And today, in human genetics, there are papers on estimating genetic correlation using genomic data. So it's quite common. This is something we have been doing in animal and plant breeding since the mid-50s. And we do it routinely. Now, another couple of important papers, of course I cannot honor every possible contribution in this long history, so I'm singling out those that I think are more important or more relevant to my interest. So two persons, Clark Cockerham and Oscar Kempthorne. Kempthorne, a statistician, Cockerham, initially an animal breeder, but then became very famous when he moved to North Carolina State and published papers on population differentiation, FST statistics, and also decomposing non-additive variability, gave a general decomposition of non-additive variance into independent components. He used the term orthogonal components, such that when you have the total variability, you can decompose it into an additive variance, dominance variance, additive by additive variance, dominance by dominance, additive by dominance, etc., 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 depending on the number of loci involved. But this decomposition holds when there is no selection, no inbreeding, no assortative mating, no mutation, no migration, and linkage equilibrium. Linkage equilibrium is a word that tells that the segregation, not the segregation, but if you observe the genotypic distribution at locus 1, it doesn't tell you anything at all about genotypic distribution at locus 2 beyond what the marginal distribution at locus 2 would tell you. Okay? So all these assumptions are, of course, are violated in practice. Actually, linkage equilibrium is a word that you hear and read everywhere in connection with something called genome-wide association studies. So if you believe those assumptions, you believe in the tooth fairy. Now, this theory works. This is just one of the many examples I could have cited. These are trends in dairy cattle milk productivity for 4 major regions, US, Canada, Europe, and New Zealand. And the mean levels differ because of the production systems. In New Zealand, cattle production, dairy cattle production is largely pastoral, whereas in the US and Canada is high input, we use a lot of grain. And there has been a lot of change in milk production per cow. It's not unusual to see cows today that produce 20,000 pounds of milk, if not more. Okay? And statisticians, of course, have estimated that 60% of this change has been due to genetic improvement. Genetic improvement that can be capitalized by use of technologies such as artificial insemination. So it works. I'm going to review very rapidly some of the main statistical methods that have been used in animal breeding. Well, another important contribution to Pearson was how from data you can estimate distributions. There is an incredible paper on families of distributions, also early in the 20th century. Then Fisher in 1918 and Sewell Wright arrived to very similar results using different techniques. Wright invented an approach called path analysis, which was a precursor of structural equation models. And for this reason, Wright, I think, is the only geneticist that was made a fellow of the American Economic Association. Not that that's a great honor, given the reputation of the economists, but so be it. Then the field became more statistical. The connection between least squares and levels of variance became more obvious. The way that least squares could be applied to non-orthogonal data also became quite clear. Then in models, in linear models or nonlinear models, we have some terms that are called random, and they have a distribution, and you're interested in estimating variance components. So methods of variance components were suggested, such as Henderson 1, 2, and 3. Then a famous statistician from India, from Calcutta, migrated to the United States, Kalyampuri Radhakrishna Rao, and he suggested a method called minimum norm quadratic unbiased estimation, a minimum variance quadratic unbiased estimation. Then Henderson, I will talk a little bit more about that, coined a term that he called best linear unbiased prediction, and this actually was the most important contribution of statistics technologically to animal breeding. Then maximum likelihood methods for variance and covariance component estimation became computationally feasible, and a lot of software was developed for that purpose and still used, like ASREML and DMU. DMU is a very powerful package that was developed in Denmark. And then when the Markov chain Monte Carlo methods came into the picture and animal breeders migrated a bit away from the strictures of the linear model, some other things happened, like development of methods for order categorical data, for survival, such as in the medical sciences, using mixed effects model for Weibull hazard functions, similar things to that. Then there was a search for this unknown and elusive quantitative trait loci. And naturally, with the advent of genomic data, we have joined the bandwagon of genome-wide association studies, whole genome prediction, and also non-parametric methods. I will talk more about genome-wide association studies. I'll talk more about whole genome prediction. I will talk a little bit about non-parametric methods because I have made a bit of transit in these areas. As Professor Xu pointed out, I have been involved with suggesting Bayesian methods in animal breeding in the early 80s. I was very lonely at that time because nobody liked them and they didn't understand them. So Bayesian methods have played a very important role. I think in the field of statistics, the main instigator was a couple of papers by two econometricians called James and Stein that show that any time that you estimate a linear model with more than 2 parameters and you use maximum likelihood, you are using what is called an inadmissible estimator. Okay? You can produce a nonlinear estimator that is better in the mean square or sense without the parameter space. So some people started questioning foundations. The field became very confrontational, very philosophical, and the Bayesians made a lot of inroads, but mostly because computational methods became available, actually developed by physicists and people working in image analysis. Two notorious names are Metropolis, the inventor of the Metropolis-Hastings algorithm. It's not the city where Superman lived, okay? It was a guy called Nick Metropolis, and he lived in Chicago. And then Geman and Geman that had a paper in 1984 in image analysis. And the statisticians, the Bayesian statisticians said, hey, that's the answer that we need to do all these highly dimensional integrations that we preach about it, but we cannot do. So essentially, you use the Bayesian formalism, you invent a prior, you combine it with data, and you get something that we call the posterior distribution that hopefully is sharper and allows you to make sharper statements about the model than you would a priori. But the problem is that most of the times the prior is an act of imagination. Depends how many bottles of scotch you have had the night before. Notwithstanding, and this has been a topic of hot debate about what happens when you introduce subjectivity in science, a lot of Bayesian methods have been developed in genetics. And here is a menu, perhaps a bit incomplete, and in some of these areas, animal breeders have made their modest contribution, including several people present in this room, such as Rob Templeman, that did a lot of stuff with thick-tail processes, if my recollection is correct. And now Bayesian methods are used for genomic evaluation of farm and animals and plants. They are not necessarily the most efficient ways of doing things computationally, but they are done, and you can fit any model that you want, essentially. So let's go back to this problem of unraveling complexity. And I'm going to oversimplify and describe three main paradigms that are in use today. First, let's address this question. Mechanistically, we would expect that interactions are there. Okay? So then we have this dialogue or pseudo-dialogue between the persons that appreciate genomics and complex biology, and then the quantitative geneticists, that they insist that no, everything is additive, so the world is a multiple linear regression, and we are going to learn a lot from these multiple linear regressions. And there are good reasons to say, yes, the world may be locally, a multiple linear regression because of this paper published in PLOS Genetics in 2008. That no matter for a number of systems, real data and simulated data, they found out that most of the variability is actually additive. So if the system is epistatic, it behaves as if it were additive. So this is interesting and is a good practical method because it means that with additive models, we can successfully describe, we can successfully predict, but it also puts a plateau on what we can discover, because if everything turns to be additive, we are going to believe that the world is additive even when it is not. So until sometimes the epistatic guys come out of the closet, we will not be able to learn if they are epistatic or they are additive. And there is a lot of evidence that epistasis is quite abundant, but does not translate into components of variance. So the most widely used tool to unravel complexity is what is called genome-wide association studies, in which the basic idea is using markers. You try to find an association, not a causality association, between markers. The idea is to search for an association between a marker or genomic region and a phenotype. And let me give you a brief background on genome-wide association studies. So this is a pervasive approach in human and animal genetics. The first such association study was undertaken in Japan in macular degeneration. It's a disease of the eyes. And if you go to a couple of sites in NIH in the web, you'll find out what are called 13,418 curated associations, and in another side there are 44,924. And associations include studies of breast cancer, waist circumference, hand strength, erectile dysfunction, intelligence, propensity to cry. And the prevalent methodology simply uses a linear regression on a marker locus. And not surprisingly, if a trait is complex, you capture a tiny amount of the variability. The technique is most of the times ordinary squares. So if you are testing 10 million markers, you are going to have 10 million tests. And then the discussion of methodology in the literature focuses primarily on a type of link function. For example, it's not good to use a linear model when you have a 0-1 trait, for example, or in correction for something that is called population structure. It's whatever concealed heterogeneity there may be in the data, whatever it means. And of course, correction of multiple testing, because even if you have no association whatsoever, if you do 10 million tests, you are going to find something, just due to chance. So there are these so-called methods for multiple comparison errors. And this, of course, led the human geneticist through an industry of conjectures about missing heritability, which was actually an excuse to make meetings in Italy, for example, on missing heritability, or to argue from their perspective. If they were working about epistasis, they would say, oh, we are ignoring epistasis. If somebody was working with copy-variant numbers, oh, they are not taking into account copy number variance, etc., etc., etc. And of course, the agricultural geneticists laugh about this because you would never expect that a single locus would contribute a large portion of the variability for a complex trait. So in most of these genome-wide association publications, you see these plots that are called Manhattan plots, in which they scan chromosome by chromosome, they compute the p-value, some measure of statistical significance, and then after corrections for multiple comparisons, you develop a threshold, which is typically very stringent, so very few markers graduate as being an association. Now, quite often, these results from genome association studies do not translate into predictive power. And there is a paper, that's something that we have been quite aware in animal and plant breeding, but seemingly for some reason nobody makes the points with sufficient emphasis in genetics, in human genetics for reasons that I ignore. So this is a very recent paper that gives an explanation of why these things in GWAS do not translate into predictive ability, and this was recently published in PNAS, okay, by a person like Herman Chernoff from Harvard that is very famous because of the Chernoff phases. And even more recently, and first let me say, this is something that we have been telling to our animal breeding students for a number of years. This paper published in Nature Genetics states, contrary to what current narratives might suggest, GWAS have not discovered these genes because of linked equilibrium arising from historical co-inheritance of neighboring variants, and any one of a number of SNPs could be causal. So linked equilibrium really makes a mess of the interpretation of genome-wide association studies, which are just based on marginal associations. So then everybody talks about the n smaller than p, m being sample size, p the number of planetary variables. Well, we have been coping with that in animal breeding since 1948, because quite often we have many, many, many more genetic values to infer than data points. For example, we can talk about the genetic value for milk production of a bull, and in spite of biotechnology, the bulls still do not produce milk. Okay? So Henderson was the first one to address this problem. And he introduced another paradigm, so to speak, that was a multivariate extension of Fisher's model for additive effects. Instead of dealing with additive variance, he dealt with additive variance covariance matrices, where this covariance matrix is proportional to the resemblance between relatives in terms of additive gene content. So in the left, I just have a photo of Oscar Kempthorne and Fisher. This was taken in Ames, I think in the mid-60s, because Fisher used to go to Ames to teach his statistical method for research workers course. And the reason why I put this Bayesian skip out is it gives me an opportunity to remind you to tell you another story. You may not know that Fisher's daughter, Joanne Box, married George Box, a Bayesian statistician. So they divorced. So I'm sure that Fisher is, and now Box passed away, George Box passed away. I think Joan Box is still alive, and she lives in Madison, Wisconsin. So Henderson arrived at a solution that actually has a Bayesian interpretation. That's probably why it's so useful. So essentially, today we refer to this method. Animal breeders typically talk about best linear bias prediction, and we know what we're talking about. But also under Gaussian assumptions is the mean of the posterior distribution under certain conditions that you know the variance covariance structure up to proportionality. And if you adopt the more machine learning, more fashionable technique, you call a penalized likelihood using the L2 norm. So the idea is you have a linear model in which you regress on X and on Z. These are matrices that have different interpretations. And the difference between these two location vectors, beta and U, is that beta does not possess a distribution. That's called a fixed effect. And U possess a distribution, in this case, a multivariate normal. So what Henderson did is he multiplied the marginal density of these random effects times the conditional density of the data given the fixed and the random effects, took logs, and then took derivatives, set to 0, and boom, obtained a system of linear equations called the mixed model equations. That in animal breeding we call them Henderson's mixed model equations to honor that contribution. And this set of equations have been instrumental in animal breeding because it allows us the possibility of introducing all relationships among individuals, even in the globe. In dairy cattle breeding, we do an international evaluation of bulls in an organization called Interval, where we use the genetic relatedness between all artificial insemination bulls in the world that are active, at least in developed nations. And we include this in that matrix. This matrix actually can be inverted in a very simple manner. And then this can be applied either to univariate problems, milk production, or multivariate problems, milk production, fat composition, resistance to disease, longevity, etc., to the extent that you are willing to use the Gaussian assumption. And in Finland, they have developed a solver that solves up to 250 million of equations in a feasible time. Keep in mind that in animal breeding, in practical animal breeding, normally the animals are evaluated every month, for example. In pig production, actually every 2 weeks, and the same is in chicken production. These numbers may not be very accurate. So large number crunching using these equations has been something we have been doing quite routinely. But there is an interesting anecdote about this. Henderson presented this line of argument and this line of development in a meeting in the early 60s at University of North Carolina in Chapel Hill. So, and he called this solution beta had maximum likelihood and U had maximum likelihood. So the first question he had in the meeting was from Rao. Rao said that he was not maximizing a likelihood because this thing, it's not a log likelihood. It's the likelihood is the density of the marginal distribution of the data viewed as a function of the parameters. But this is actually not the marginal, it's not the log of the margin, it's the log of the joint. So, and Rao added, he said, well, in any case, let's use the word illegal maximum likelihood. So Henderson was a bit uncomfortable because being questioned by Rao was not a minor thing. Then the second question was by Oscar Kempthorne. And Kempthorne, being a brilliant statistician like he was, he realized that by doing this sort of thing, he was shrinking the estimates towards 0 in this case. And Kempthorne didn't like that because he would create bias in the standard statistical sense, like ridge regression, for example. Ridge regression is a bias estimator, but because he has a duality viewed as a best linear bias predictor, it's an unbiased predictor in the sense of Henderson. So something that is biased an estimator is unbiased a predictor. It doesn't make any sense, right? So, and there was a lot of discussion about that. Almost at the end of the discussion, somebody who's sitting in the last row, was a man called Irwin Bross. He was a statistician from Kodak Company from Rochester, and he said, well, the problem is that Henderson is using a Bayesian argument. And this thing is the mean of the joint posterior distribution of beta and U in a Bayesian model in which a uniform prior is assigned to all the betas and a Gaussian prior is assigned to all the U's. Well, being called a Bayesian in the early 60s was a problem. And Henderson told me I almost fell from the podium. Okay, then enter the genomic era. This is a schematic that you can find in Wikipedia, as you know, about something called single nucleotide polymorphism. These are markers. And you observe this is a double helix in chromosome 1, a double helix in chromosome 2, and everything is the same except at this site in which there is a polymorphism, so you can call this allele little a and this allele large a, and there have been many such SNPs that have been discovered in the world. So now in animal breeding we have massive amounts of phenotypic data and an increasing number of genomic markers, such as the SNPs. And to make the story short, this has been essentially the evolution of the number of SNPs used in animal breeding application, from breeding fish to breeding cattle. So in dairy cattle and in beef cattle, we are routinely genotyping animals with chips of about 800,000 markers. And the sequences are becoming available. Okay? We are going to need many such sequences, but the number of variants in a sequence is enormous. 24, 25 million, as per a paper that was published last year. Okay, in 2001, a very good solution of how to deal with this massive amount of data in the prediction problems was proposed, which was a paper by Mewisen from Norway, Hayes, and Goddard from Australia. And they essentially suggested to regress, instead of using the genome-wide association paradigm, regress the phenotypes on all available markers simultaneously, and that they proposed a couple of methods called Bayes A and Bayes B. They differ in the prior distribution that are assigned to the marker effects, and this paper has produced a tremendous impact in animal and plant breeding industries, and everybody has embraced the area called genomic selection, which is what we are doing today. And unfortunately, we came 2 years later to the party. This paper was essentially interested in prediction, but we also warned that if inference and invariance with respect to a set of priors used, they would suggest the data are informative enough, and that the prior is overwhelmed by the information contributed by the data, which unfortunately is not the case when n is smaller than p. Why is this so? Well, let me argue from the following perspective. In statistical applications, you're interested essentially, one, about learning about the state of nature. And for that, we call that problem inference. And we normally use the data already mentioned, and you come up with some sort of a model, and then you talk to your friends in statistics, and they say, well, what do you recommend me to do? And some people like moments, other ones like likelihood, other Bayesians. And then the theoreticians will try to convince you that under regularity condition with bounded third derivatives, etc., you will have a method that asymptotically converges to a value and in the class of whatever is best in whatever sense. And now, of course, the same course, but with different models, that you really have to search for a method that fulfills the two oracle properties. One is that you hit the true model, and second, once you hit the true model, you should be able to learn the parameters asymptotically. Great. So, but when the number of parameters exceeds sample size, the likelihood does not have a non-singular information matrix. Remember that you learned this in the first class of statistics. You didn't learn it, you should take it again. That when you have a data set of size n, the largest number of questions that you can ask to a data are n. If you ask more than n questions, the answers will be linear combinations in the class of linear models of the n answer you formulated before. So when you have a sample size of size 1,000 and you fit 25 million regression coefficients, it's quite clear that you get numbers that where is the information coming from? And it's not coming from the data. It's coming from the construct of the method, which from a Bayesian perspective is called the prior distribution. So Kempthorne that was a furious anti-Bayesian, and actually once said he said, I promise that for as long as I live, I will not allow a Bayesian in the stat department at Iowa State University, where he lost because they hired somebody called Glenn Meaden in the early 80s. But Kempthorne warned that the prior can be influential, but the Bayesians will retort, well, if n grows relative to p, asymptotically the posterior behaves like maximum likelihood. It converges in probability to a true value. But the problem we are facing in genomics is that p is growing larger than n. Okay? When you have finished genotyping your chickens with a 10,000 marker chip, Illumina calls you and says, oh, we have a sale of a new chip. If you buy it tomorrow, we'll give you a 50% reduction. So it's 50,000 chips. So you regenotype your chickens, and when you are done, another chip comes, and now you have sequences, but then they're going to tell you the sequences are not enough. They're going to measure something else. So we'll have expression data, metabolic data, methylation data, whatever. Okay, so p is growing faster than n. So I don't think the p fixed and n relative to p grows is a paradigm that we can use very usefully. Okay, so I warned about this problem in a paper in 2013 because everybody is now proposing these outlandish mega Bayesian models with mixtures. Nobody knows how the mixtures behave in high dimensions when implemented by Markov chain Monte Carlo methods, and they're making statements about the state of nature. Prediction, on the other hand, is a different ballgame. Another paper that I discovered more recently, and that gives conditions for a specific linear model, which is the ridge regression model or a model with random effects, or Marcus gives conditions that essentially agree with what I stated in that paper. So got it, okay? So when p is larger than n, one should tank these spirited attempts at inference and to argue about genetic architecture that essentially what you get is from some Mickey Mouse priors that you have invented that if you tell them to a molecular biology, they will laugh in your face. Okay. So I already pointed this out. And as a note, this term is one that I abhor because it's a buzzword that is used to get grants. Okay? And they do not use genetic engineering because it's no longer available. Genetic engineering was appropriated 30 years ago. So I really prefer to use about the statistical genetic architecture of trait, not about the genetic architecture, because I think these models are to genetic architecture what, let's say, 10 tons of bricks are to the Bilbao Gehry Museum or to Norman Foster's Bank of China in Hong Kong. Now, when you go to prediction, anything goes because you can invent any outlandish prediction machine and you can calibrate it in cross-validation. And if you do your work properly, you can replicate or pseudo-replicate your cross-validation, obtain some sort of uncertainty about your predictions. And you don't care very much about where things are coming from because you gave up. A priori, you surrender to complexity, but then you exploit complexity. And to a large extent, this is the philosophy in machine learning. You try to see how a system behaves, and then you replicate how the system behaves without necessarily understanding it. So in prediction, especially if you use Bayesian method, the objective is to develop what is called the predictive distribution for a particular model that you propose. This predictive distribution takes into account the uncertainty about the model parameters using the Bayesian logic. And then if you have several competing models, you use what is called Bayesian model averaging, in which you average predictions over models by taking into account the uncertainty associated with each of the models. But of course, if you fail to specify the right model or the right set of models, your predictions may not be good. So this is an interesting, to me, statement that I found in a book called Predictive Inference by Geiser. And let me read it to you, because I think it has a lot of meaning. Clearly, hypothesis testing and estimation, as stressed in almost statistics books, involve parameters. This presumes the truth of the model and imparts an inappropriate existential meaning to an index or parameter, inferring about observables more pertinent since they can occur and be validated to a degree that is not possible for parameters. For example, if I say heritability is 0.25 and my estimate is 0.35, I will never know if it is a good or bad estimate because first, I cannot observe the true heritability, and secondly, because heritability does not exist. It's an abstraction. Okay, so the third paradigm is machine learning. It's essentially non-parametric, and I started transiting this area in a very naive manner. I'm an agricultural scientist, okay? I'm interested in statistics and things, so I don't know very much about it, but I saw that it was very useful. And we have had experience with a couple of things, which I'll mention in a minute. So it's very difficult to define machine learning and away from data mining and away from statistical learning, which is another term invented by statisticians because they were concerned that the computer scientists were kind of winning the battle, so to speak. And it's a collection of techniques that involves things like neural networks, pattern recognition, boosting, bagging, Bayesian nets, a lot of stuff going on, and essentially are based on cross-validation. Maybe there are other ways of calibrating this. And we have had experience with essentially two types of techniques. One is regularized neural networks. We thought that it will provide the solution to the problem because they can be shown to be universal approximators, but unfortunately they are very greedy universal approximators, so that quite often when you train a neural network, it copies the training data too well, they overfit, and they predict very poorly when confronted with new cases. And the second type of technique that we have experimented with a little bit is called reproducing Kernel Hilbert spaces, mixed model regression. I was actually, I think one of the leaders in this area is Grace Waba from the University of Wisconsin-Madison, and she gave the solution to this problem using calculus of variation. It's a technique widely used in physics to find functions of anything. For example, this could be a sequence for an individual animal, or could be a string, or could be a partiture if it were a musical problem, and then you try to navigate in a Hilbert space of functions and find the function that is optimal in some sense. And the mathematics is, of course, completely beyond me, but I saw that this was something that had some potential. And I may be accused of doing cherry picking, but what I'm going to show is not atypical of what it has been found out in practice. This is a paper was published in Crop Science by scientific staff of the United States Department of Agriculture at Cornell University, where they had data for 18 trials involving several species such as barley, wheat, maize, etc., etc. Different sources, different number of markers, and they fitted a number of, let's use the term prediction machines. Some are Bayesian hierarchical linear models, other ones are machine learning techniques such as support vector machines or reproducing Kernel Hilbert spaces regression. And let's concentrate on this row here, which are called the predictive correlations. Here the data are divided into training and testing sets. You fit the model in the training set, and then you predict the masked data in the testing set and compute the correlation between prediction and realized value. And if you observe, essentially there are very little differences between prediction machines. There is a 0.41 here. That's probably a mistake, or maybe they implemented the support vector machine incorrectly, because the support vector machine is supposed to be one of the state-of-the-art things. So nothing to go to war about it, and this is something that you observe in practice, that quite often the prediction machines, at least applied to animal and plant breeding, do not differ very much. And also, if you measure cross-validation, the cross-validation uncertainty is always much larger than the difference between methods. So it's hard to conclude one thing or another. Now, do you like malt whiskey? Okay, so here are some malt whiskeys that you buy in duty-free shops. Okay, they are not the top ones. Some are very drinkable, like this one here. This, I cannot read it, but that's pretty good. Glenfiddich is, and it's so pale that you get depressed before you start drinking it. But unless you are trained on how to drink whiskey, you probably will not note differences. You can see difference in color, sometimes in taste, but you don't know how to. So let's use whiskey secrets. Okay, the whiskey secrets are you have to replicate the comparison. Careful with whiskey, okay? Don't replicate too much. It's a very addictive drink. And also use paired comparisons. That is something that Gustavo and I have been teaching a prediction course in different places, and we try to emphasize that when you compare prediction machines, if possible, you should do a pair prediction. So I'm going to show you something. I took this data from the paper in Crop Science. There are 18 trials involving different species, and let's do a comparison, a pair comparison between the results of these trials, and choose two methods. One method is reproducing Kernel Hilbert spaces regression, and the other one is called Bayes-CPI. It's a variable, it's a Bayesian, quote-unquote, variable selection model in which you use a mixture in the prior distribution. You mix a zero state with a Gaussian, and then the mixing probability is treated as unknown. And these are the results. There are 18 comparisons. This is the tie line, so to speak, and there are 16 victories in terms of predicted correlation for reproducing Kernel Hilbert spaces regression, and 2 victories for Bayes-CPI. So if you use a soccer metaphor, this is a game like Hilbert won 16-2 versus Bayes-CPI. But if you look at the marginal predicted correlation, you didn't see any difference. You wouldn't go to war about it. So if you believe that in the last World Cup in 2014, the Germany 7, Brazil 1 result was a decisive victory, you'll have to conclude that this is an even more than decisive victory of the non-parametric approach. Now, I am not doing cherry picking because it's not my work. Maybe I'm cherry picking in putting a study in which this comparison was made, and Hilbert, reproducing Kernel Hilbert spaces regression came victorious, but this is not unusual. That you look at the prediction machines and they don't differ very much, but when you go and look at it in detail in terms of pair comparison, if you can, you start seeing differences. A group from the University of Chicago, and I think Ana Vasquez has also worked in this front, have migrated in the question of using whole genome prediction in medicine, which is something that's not picked up much yet. Because in personalized medicine, you need very precise prediction. Like, for example, in chronic lymphocytic leukemia, there is a protein called CD38, another one called SAP70, and some mutations in the immunoglobulin heavy chain region that have very strong predictive power. And the whole genome approaches applied so far have not conveyed an equally strong predictive power. But if you augment the battery of predictor variables with what is called polyomic information, such as expression data or metabolic data, you can enhance your predictions. And we have recently, and by the way, this paper uses something called omic kriging that is also a special case of reproducing Kernel Hilbert spaces regression. And we recently used methylation data from a plant called Arabidopsis thaliana. There were 123 isogenic lines. These lines were genetically identical, but they differ in their methylation profiles. It's called epigenomics. And there were 740,000 probes So this was a question in which P amply exceeded sample size, and we compared different implementations of reproducing Kernel Hilbert spaces regression, and the use of methylation data gave pretty good predictions of plant height, plant height or flowering time. I don't remember. Arabidopsis is a very tiny plant like this. So getting to the end, we started a bit later, and we have this interruption, so I'll allow myself to have a few more minutes if you don't mind. If you have to go, please feel free. Let me talk about some of the challenges that we face, I think we face in, at least in ALMOB. Well, first, sequence information. Instead of SNPs, we're going to have 25 million markers, at least in couple. Then the data will continue growing in the sense that I pointed out before. Then there is the question of discovery and causality that everybody talks about causal variance, causal variance, causal variance. And they say, well, because we have a sequence, all the causal variance will be there. Well, I think we will need many good courses of causal inference in the training of biologists, because to establish cause is a really involved process. It's like saying that a microorganism is responsible for the disease. Microbiology 100, bacteriology 100, Koch postulates. You have to go to a sick person, isolate the pathogen, clone the pathogen, insert the pathogen in a healthy person, produce the syndrome, and then isolate the same pathogen again. And then you establish the cause-effect relationship and a causal argument. But now, since people cannot do randomized experiments, they have developed method for learning causality, and they seem to work, but one needs to be a little bit careful about it. And the methods are growing in dimensionality because we have more variants, and the problem will get intrinsically more difficult. So if you get into the business of making this family of Bayesian linear regression methods more ambitious, larger, etc., you will find out that the more variables you have, the regression coefficient will have to become smaller as you increase the dimensionality of the problem. It's very simple, because if you have a sample size as 1,000, it's like having a pizza for 1,000 people, but then you invite 25 million persons to eat the pizza. You have to divide the pizza in 25 million pieces. And that's what the shrinkage method do. So as P grows faster than N, the N by N method, so to speak, become more interesting computationally whenever you can apply. Another challenge I think we have is this new approach that is called systems biology, system genetics, which I will argue that it will not be very useful in the absence of rate coefficients. Most complex traits require measuring some rates to understand things. So I'm not convinced that this will have a very strong power, but I may be wrong. After all, the reductionist approach has served us beautifully. And then the other problem is that quite often these systems are learned from bacteria, from flies, and as far as I know, the pigs do not fly yet. Maybe if you do some gene editing, we'll be able to make some pigs to fly, but we are not there yet. Conclusions. After these fast passes through history, personal anecdotes, and personal opinions, and I hope you excuse me that I've taken the liberty of expressing my mind, animal breeders, I think, have taken up statistical ideas rapidly. They not only have uptaken these new developments in the field of statistics rapidly, but they all have also made contributions of their own, such as best linear bias prediction, notoriously Henderson, this method called REML for variance and covariance component inference, Bayesian modeling, Bayesian linear regressions, and more, and today, genome-enabled prediction. Our main science is called quantitative genetics. It's a naive theory, but it's quite simple and very powerful, descriptively and predictively. But I don't think it's very powerful from a learning perspective. It may be useful. It's, after all, statistics didn't they say, is organized common sense. So maybe we can organize our common sense even better. Inference and prediction are connected, but they're very different. You can have a model that may have very, very good predictive power, but not be good to explain the state of nature. And quite often, you may have a very theory, but it's quite complicated and may be true in some sense, but you use a simpler model, and the prediction may be better, because a simpler model is robust. So arguing predictively, going from the good predictions to a state of nature is also something that requires a bit of care. My prediction is many of these GWAS will be G-washed away. And then this parametric method that I've been using in quantitative genetics for many years are really challenged by the genomic and post-genomic data. And I have been saying this for a number of years. I would like to believe that there's going to be an analytical shift towards semi-parametric and machine learning type techniques, but the animal and plant breeders are still very married to the notion of additive genetic value, which has served us quite well. But curiously, as far as I know, there has not been a very severe and stringent test of this theory in a randomized experiment. We can always argue observationally that because we have used the additive genetic model, we have made progress. Well, that's an assumption. Maybe if we had selected animals in a different way, using different ways of smoothing the data, we could have made an equal amount of progress. That I cannot answer because we don't have randomized experiments that could answer the question unambiguously. So I'll leave here with a statement of Mark Twain that it ain't what you don't know that gets you into trouble, it's what you know for sure that just ain't so. Okay? So thank you very much for being with me today. Thank you for the invitation. Thank you all for hosting me at Michigan State.